Good morning students, I am Banerjee. I am working as a TCT teacher in MJP DCWR school, Samanasa. Okay, so today I want to tell about the cell. So the topic is cell the basic unit of life. Cell the basic unit of life. So our earth is a beautiful place where in different living things coexist. Different living things coexist. Next, so these are from mosses to huge coniferous and also invisible bacteria to huge blue whales, a big blue whales. Now these all organisms have a specific functional unit is called cell. A specific functional unit is called cell. Now let us study about the cell. So before the first microscope was invented in 365 years ago. 365 years ago. The people were not aware of the living world, about the living world. That is invisible living world. They don't see the naked eye the invisible living world. So many scientists are observing or describing about the invisible microbial world. Then with the help of microscope, among them, so there are 4 scientists are working or worked about the describe of invisible microbial world. There are, first one is Athanasius Kircher. Second one is John Swammerdam. Third one is Antony von Leeuwenhoek. Antony von Leeuwenhoek. Fourth one is Robert Hooke. They are worked and observed the invisible microorganisms. So here we need to tell about the Antony von Leeuwenhoek. Antony von Leeuwenhoek was the first person to see the bacteria, protozoa and red blood cells. So Antony Leeuwenhoek was the first person to see the bacteria and protozoa, red blood cells and teeming living cells in a drop of water. So and also he used different lamps to describe or to understand the microorganisms, invisible microorganisms. Okay, you may recall all the living organisms carry out certain functions. The sets of organs perform specific functions. What is the basic unit of a organ specific function? What is the basic unit of organ? The specific function. Here we need to know about the structure and we need to know about the structure of cell. So then we need to understand the microscopic and microscopic slides. So these are helpful to understand the invisible microorganisms. Invisible microorganisms. First, the discovery of cell. The discovery of cell. So, in the year 1665, so a scientist, Robert Hooke, a British scientist, he observed the slices of a car. Slices of a car. He observed the slices of a car under the simple magnifying microscope which was made by Robert Hooke. The car is in oak tree. It belongs to oak tree. So that he observed the car resembles the structure of honeycomb. It consists of so many empty spaces like structures. He observed Robert Hooke. These structures made up of small cavities. Small cavities like structures. So these small cavities, Robert Hooke named as cell. What is the meaning of cell? The cell, the word derived from Latin word. Means a little room, an empty space or a little room. Now let us do the activity. Activity 1. Activity 1. Observing the matchstick. Observing the matchstick. So it will be difficult to observe the cell in the car. So as seen Robert Hooke. So Robert Hooke proposed to understand the structure and to see the cell in a matchstick. So here, take a matchstick and soak in it half an hour. Then it break and take the slices of matchstick. So take thin slice of matchstick. The slice put on the slide. Then so put a drop of water on the slide. What you have collected. The slice of matchstick, then so cover with a cover slip. So then the slide kept under the microscope. So then observe what you have observed. What is the structure of the matchstick slice under the microscope. Okay, when we see through the microscope the matchstick structure the matchstick slice of the matchstick structures. So looks like a small spaces or rectangular spaces. It is similar to the practical done by the Robert Hooke. So what is the triangular shape of, what is the triangular shape of spaces called? This called cells. These are called cells. Okay. Then it is very wonderful, and it was a milestone. So to know the cell discovered by Robert Hooke. So the cells are the cork cell, and matchstick cell. Cells are dead cells. Dead cell. Okay. Can we see the living cells in animals or in plants? So if we can we see the living cells in organisms? So we can do practical on the living cells, how to identify or how to see the living cells in living animals or living organisms. So let us try to do the activity 2. Observing the onion peel. Okay. So an onion peel. Take the fleshy portion of the onion bulb. So break into two pieces. Break into two pieces. Then break the two pieces. You will see the translucent membrane in between the two pieces of onion peel. The translucent membrane. We will see the translucent membrane in between two pieces of the onion peel. Then take a small membrane. So the small membrane put on the slide. Then put a drop on the slide. The cover with the cover slip. So observe under the microscope. Then the rectangular spaces like cells are seen in the microscope, through the microscope. Now here these are called onion peel cell. This is called plant cell. This is in live condition. This is called plant cell. It would be similar to the cork cell or matchstick cell. Can we see the living cells in animals? So if you know that thing, let us do activity 2-3. Activity 3. Observing cheek cells. So now we will see the living cell in human beings. How to see our cells, living cells in human beings. Then, so we will do the practical or activity. So then, take first of all, so wash your mouth with water. Then scrap the take a wooden spoon or plastic spoon. Then so you take scrap the inner surface of the cheek, inner surface of the cheek. While you are doing this experiment, you should care about. So don't too much scratch the inner surface because it may be injured. Then so take the extract of the cheek, extract of the what you have collected cheek material. So place on the cover slip, placed on the slide, and drop a water. Put a drop water. Then so cover with the cover slip. So then observe so you may observe under the microscope. Then you may observe under the microscope. So you will see the cells through the microscope. Here, so the cells are. So is there any similarity to the onion peel cells? So is there any outer surface? Here, so onion peel have a outer surface, a thin outer surface. Here, human cheek cells have no outer surface. Then a significant observation. A significant here, Robert Brown, one of the scientists, he made a contribution to understand the cells. Robert Brown. He made contribution to understand the structure of cell, to understand the cells. Here, so many scientists. So there is a small spots in the center of the cell. So they are called Pili spontana. Pili spontana and other scientists, they first this saw the nucleus. They saw the nucleus in the cells. He observed in the epithelial cell of the animal. He found the nucleus in the epithelial cell. Epithelial cell. Then, so every cell have so a specific function, a specific spot-like structure. Here, so Robert Brown so named that spot that is called integral part of the cell. He said that so nucleus is the integral part of the cell. Integral part of the cell. Integral part of the cell. Named them as, so Robert Brown named this part as nucleus. Named the part nucleus. So the nucleus is a spot-like structure. Appear is a opaque and in between the surrounding cells. So and also Robert Brown found that a small apertures, a small pores, these are called stomata. 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 The stomata are useful or helpful. The stomata